So good morning, everybody. It's my pleasure to welcome you on this session on the room number 2. And the session will be about metaomics and microbial genomics. So we will have the pleasure to listen to three, in my opinion, very interesting talks to come. First, I would like to present Clémence Frioux. So Clémence, she's from Bordeaux. From INRIA Bordeaux, and she will present a work that was published last year or this year, I forgot, in the Journal of eLife, which is about the metabolic complementarity between microbes in the way of understanding how these microbes shaped community. So, Clémence, the floor is yours.
Good morning, everyone. I would like first to thank the organizing committee of JOBIM for the opportunity to talk. I will present methods dedicated to assess the metabolic complementarity and identify key players in microbiomes. Microbiotas are surrounding us in our body, but also in the ocean or in the soil. They consist of thousands of species that interact in a shared environment. These species, among which many have never been cultivated, form a complex network driven by these interactions occurring between members. Interactions are largely mediated by metabolites that are both produced and consumed by all of these species. Therefore, the diversity of functions in a microbiome is a key to understand how these ecosystems live or react to perturbations. Most microbes cannot be isolated, so omics experiments are the main source of information. In the past, we mainly wanted to answer the question who is there using amplicon sequencing, but now shotgun metagenomics becomes routine, enabling us to get all sequence from a sample and answer the question what might microbes do in this particular ecosystem. We can reconstruct genomes from metagenomes that are called MAGs. And with that, it gets possible to apply similar methods to those applied to individual genomes. One of them is the reconstruction of metabolic networks that I will discuss today. A metabolic network is a set of metabolic reactions, like this one here, transforming a substrate A into a product B. Metabolic reactions are catalyzed by enzymes, here. And at genome scale, a metabolic network actually contains all reactions associated to any enzyme found in the genomic sequences. Usually, that makes a set of 1,000 to 5,000 reactions. All these reactions are the functional repertoire of the cell. What could an organism do? We are in a context of communities of organisms living in a shared environment. Let's assume we have a genome or a MAG for each of them, so each of our two bacteria. What we would like to do is build a metabolic network at genome scale for each bacterium. Using these metabolic networks, we could identify complementarity between the metabolism of each species and suggest possible cross-feeding interactions. Here, the green bacterium produces D, the blue one consumes D. This could be a cross-feeding interaction. And in case we are interested in a given function, here G, we would like to find key species, key players that are associated to that function. Here, clearly the blue bacterium, but also maybe the green one because it can produce D that is needed by the blue one. So we can't find these interactions just by looking at the metabolic networks. They are static objects, and if we want to simulate metabolic behaviors in specific conditions, specific environments, we need to apply mathematical frameworks to our graphs. Here, we chose a qualitative modeling called network expansion that recursively computes reachable or producible metabolites starting from a set of available nutrients. Here, A and B being nutrients, C, D, and E are the set of reachable or producible metabolites. We implemented this algorithm in answer set programming, ensuring scalability to a large number of metabolic networks, and we used it in our pipeline that I will now present to you. This pipeline is called MetaJ to Metabo, or M2M, and I will show you its main steps. Starting from a collection of annotated genomes, or MAGs, M2M automatically reconstructs metabolic networks for each of them. We use pathway tools and the Metacyc database to do that, and we actually built a Python wrapper to ease the automation. Our first analysis then consists in establishing and comparing the individual metabolic potentials of each microbe in its environment. But we know that organisms actually interact with each other. So we compute the collective metabolic capabilities that take into account the complementarities between metabolic networks. This is actually putative cross-feeding interactions that we take into account at that step. That way, we can identify metabolites that are producible only through mutualistic interactions and can therefore not be produced by a single microbe. Here are the green and yellow stars. These metabolites, or any others, can be set as metabolic targets for minimal community selection. The objective is to determine communities that can produce compounds of interest, so our green star or our yellow stars. Altogether, this pipeline, M2M, aims at systematically screen the metabolic potential of microbes and the mutualistic potential of a large community. The main step of our pipeline is the selection of minimal communities. This is a combinatorial problem solving, reducing the complexity of an initial community of species, living in an environment where nutrients are available and with a metabolic objective that is producing a set of compounds. So we can find a minimal community, but it's very likely that one minimal community will not be the only solution to the problem. Much more likelier, we will have a lot of equivalent minimal communities, up to millions, because the metabolism is highly redundant. So we have a huge combinatorics to deal with, and enumerating all minimal communities as well as analyzing them is very demanding. So instead, we propose the concept of key species that can be very easily computed and is the set of species found in at least one of the predicted minimal communities. So here we have 4 optimal minimal communities, but altogether only 5 symbionts occur in them. And we can further distinguish key species into 2 categories: essential symbionts that occur in every minimal community, like the yellow symbiont, or alternative symbionts occurring in some but not all communities. As it is very efficient to compute, we believe that the key species concept addresses the combinatorial challenge brought by the functional redundancy observed in natural communities. We applied our pipeline to a collection of 1,500 reference genomes from cultivable species of the human gut microbiota. We built metabolic networks for each of them and looked at metabolites that could only be producible through cross-feeding. We classified these 156 metabolites into 6 categories: lipids, sugar derivatives, etc. And we made them metabolic objectives for minimal community selection. For each group, we computed the key species among the initial 1,500 species. but we also enumerated all equivalent minimal communities to evaluate the combinatorics of the problem. So here we have our 6 categories. The minimal size of a community is ranging from 4 to 11 bacteria. And you can see that we have up to 7 million equivalent minimal communities for sugar derivatives. Fortunately, the number of key species is way smaller. For example, 185 species out of the 1,500 for lipids metabolites. And the number of essential symbionts is even smaller. We studied how these key species interact and connect with each other in communities by studying the composition of every enumerated communities. It's very hard to represent 58,000 of 7 million equivalent communities. So we built association graphs and compressed them using power graph analysis. So that way we compressed clicks and bclicks motifs in network, ending up with representations like the one you have here. So small nodes are species, key species, and large nodes denote groups of species with an equivalent role in the communities. They were compressed during power graph computation. For the example of lipids, we have 58,000 communities, and each of them is composed of one species from the node 1, one species from the node 2, this species from node 3, which is an essential symbiont, one species from node 4, and the 3 species from node 5, which are also essential symbionts. In addition, when we looked at the taxonomy of the species, we observed that groups of equivalent bacteria in large nodes here usually share the phylum, suggesting that they carry functions that only specific members of the phylum also carry. So here, for example, lipids production needs a function carried by this group of Bacteroidetes. This is a reading pattern that we observe for all groups. And when we went back to biology, we observed that essential symbionts were sometimes shared between several groups of metabolites. And we found that some essential symbionts are actually already known and studied in the context of probiotics. So the first experiment I showed you was performed on reference genomes, but MAGs are the main genomes available for microbiomes, and we know that they are possibly incomplete or contaminated. So we designed an experiment to assess the impact of MAGs incompleteness on metabolic network reconstruction and subsequent modeling. To do so, we removed between 2 and 10% of genes in all or subset of our initial MAGs. Then we ran our pipeline. Regarding metabolic network reconstruction, so we observed, as expected, that the number of genes in MAGs is lower than the average number of genes in reference genomes. But this cannot be seen as after metabolic network reconstruction. They all have similar numbers of reactions, pathways, and metabolites. So that's good news. What happens when we model the communities? There is a strong metabolic redundancy in microbiotas, and this reflects on a very stable set of compounds producible by communities, regardless of the degradation of the genomes. Some effects are more visible when we look at key species computation. There, the more we degrade the genomes, the more different are the sets of key species. But we can see that for small degradations, the set of key species is relatively stable. So we can conclude that the criteria typically used for MAG quality, which is more than 80% completeness, is likely to be sufficient to get a coarse-grained yet valuable and reliable first picture of the metabolism in a community. As a summary, MetaJ2Metabo is a flexible pipeline for large community analysis at the metabolic scale. It's very flexible. You can reconstruct metabolic networks in the pipeline or provide them as input. Each step can be run individually. The purpose of the pipeline is to get a first picture of the metabolic complementarity and metabolic redundancy in a large microbiota. Its second objective is to reduce the complexity of the initial microbiota by pinpointing members of interest for targeted functions. In my presentation, I illustrated the use of M2M for genome or MAX collections. However, another use case that is presented in the paper is to build a community by sample in a metagenomic cohort, one sample, one community, and then compare them. So we did it for samples in the MetaHit dataset. Finally, just to remember that we look at might be producible, and we would need metatranscriptomics to further refine predictions in the context of each environmental condition. With that, I would like to thank you for your attention, thank all my colleagues and collaborators, and let you know that we are hiring postdocs in RIA Bordeaux. Thank you.
Thank you very much, Clémence. Don't see any. So again, if there is any question, don't hesitate to ask them in the chat. But before them, I could start with I have plenty of them, but I will start just maybe with the previous one. You were showing a very interesting result to me about the especially the MAGs and the fact that you could use the M2M pipeline to better understand the quality of the MAGs and something like that. Could you have any? Could you give us any idea about? In my opinion, it could be a bias because of the metabolite that you are looking, because some of them they are associated to pathways that are very well known versus others that are less known. And could you say a little bit about if to you could use this pipeline as well to, let's say that for some pathways that are very well known, I think your approach is very cool, but also to emphasize the pathway that need to work on, because I don't know, or if in your RD there is a ranking of the pathway that you could trust versus the one that you cannot trust at all, and especially you were talking about different kind of metabolites, and maybe using your pipe, you see maybe different kind of results. I don't know if you could give us some idea about what is behind the scene at some point.
Okay, so on that topic, we highly depend on the initial database that was used for metabolic reconstruction. So the wider the database, and by wider I mean containing a lot of reactions, even those that belong to secondary metabolism or very poorly known functions, the better the metabolic network reconstructions and the predictions. So to precisely answer your question, I'm not sure we can really look at the pathways that are better reconstructed because they are better known and compare them to the pathways that are less known. But we could definitely look at the pathway composition of the metabolic networks and see for each species whether it's more complete or less complete when we degrade the genomes. So that we could do. But then we really rely on metabolic network databases to get poorly known functions.
And so I take my shot for another question because I don't see any. Sorry, I'm not necessarily used to that very much, but anyway. And about the number of solutions that you have, do you have? Maybe I missed something during your talk, but do you have the same complexity of solutions based on the metabolite that you were looking at? You were comparing the lipids, the carbohydrates, and so the compression graph that you were showing. Do you have a different complexity based on the metabolites of interest?
So no, because we ranged from, I think, 1,000 solutions for one particular group of metabolites to 7 million of solutions for another group. So there are several explanations, possible explanations. The first one is, of course, the number of metabolites that you put in your metabolic objectives. And then whether these metabolites occur in very rare pathways that are present in very few members of the community, or in pathways that are, I don't know, maybe widely present in all bacteria tests, for example, then you increase your combinatorics. So it's not necessarily related to the type of compounds you look at, but rather really the structures of your metabolic networks.
Okay, thank you. Okay, so I think we have to move on, but again, if people are too shy, don't hesitate to type your question on the chat. Clémence is still there, he could answer the question in private. Sure. A question from Samuel Chaffron. So I don't know if you, Clémence, you could see the question. So you talk about the objective function for identifying minimal communities. Could you give more details about what are these functions and how you select them or how you build them?
Yes. So maybe some of you are familiar, and Samuel is certainly is, with objective functions in flux balance analysis. So in our case, objective functions are way simpler because we are using qualitative methods. And an objective function is simply a set of metabolites that we are interested in producing. So in the examples that I showed, it was, for example, a set of carbohydrates. So we just put these carbohydrates identifiers, and then the community selection will be performed to ensure that these carbohydrates are producible in the reduced communities.
Okay, and we take the question of Roman as well. Is it possible to integrate the temporal aspect to M2M? Because ecosystems, I mean, there is a temporality at some point, and so could you introduce that in your pipeline?
So far, not really, but in a certain way, because if you are looking at longitudinal studies for cohorts of individuals, you can in some way take into account temporality by comparing the metabolites and the communities at each step, but it's not taking into account temporality otherwise.
Okay. Thank you again. So we have to move on. It was very interesting. Thank you again, Clémence. Again, if there is more questions, don't hesitate to use the chat, and Clémence could answer there is another one left in the chat, Clémence, if you could answer it. Excellent. So the next speaker is Benoît. So Benoît is from Maillage in Paris-Saclay. He will discuss about the comparison between different sequencing, metagenomic sequencing. So to compare the shallow depth metagenomics compared to the more standard whole genome sequencing. So Benoît, the floor is yours.